Hello, my name's Richard Gomer, and I'd like to thank you very much for inviting me to give a talk. I'm going to share a screen real quick. And let's see, let's take this to. Okay, so I'm going to be telling you about potential therapeutics for lung diseases. I'm a professor in the Department of Biology at Texas A&M University, and I'd like to thank especially Dr. Vaishali Chaudhary for organizing this meeting. You guys have a great program. A graduate student, former graduate student in my lab, Tejas Kahardkar, was an undergraduate at the N.B. Mehta Science College, and he clearly got an incredibly good education there. And I want to thank you for educating such good people such as Tejas. Okay, so before I start, I've got to do a couple of disclosures. I'm a co-founder of, and I have equity in, receive royalties from a company called Promedier. It's now been bought by Roche. It's developing some of the stuff I'm going to be talking to you about as a therapeutic. I'm also a co-founder of Procia, a company developing NU3 inhibitors for a variety of diseases. This is entirely work from Tejas, who was the undergraduate at N.B. Mehta Science College. So take everything I say with a grain of salt, and please do not make any investment decisions based on stuff I'm talking to you about. So the outline of the talk is I'm going to tell you about a cell type called a fibrocyte. I'll tell you how we accidentally found that there's a blood protein called serum amyloid P that inhibits fibrocyte differentiation. I'm going to tell you about how SAP is a possible therapeutic for a whole bunch of diseases called fibrosing diseases. I'll tell you what fibrosing diseases are. And then I'll tell you a little bit about the receptor for SAP. SAP acts like a signal, and how we found some SAP mimetics, and how these could be a second generation set of therapeutics for fibrosis. And then that's going to be the end of my talk. And then I'm pretty sure Tejas is going to be talking to you. He has the next part of this story. And so please tune in for his talk, because one, he gives better talks than I do. And two, his stuff is really cool. Okay, so as an introduction, when you get a little cut on your skin, there are cells called fibroblasts that are just in the tissue, and they will start proliferating and trying to work to fill in that wound on your skin. But in addition, there are blood cells, white blood cells called monocytes that come out of the blood. They go into the wound bed, and they'll differentiate into fibroblast-like cells. Now, fibroblasts and these fibrocytes can kind of help pull the wound closed. They're motile cells. And the cool thing about the fibrocytes is, for a wound, especially a large wound, is you can get a lot of these monocytes from the spleen, from the bone marrow, just from circulating in the blood. And you can actually start dumping a lot of material into the wound to fill it in, so that the wound can be filled in, and then you can lay down some nice new skin over that wound. So these fibrocytes, they start out life as a white blood cell called a monocyte, and they can differentiate into this wound healing cell, and they secrete extracellular matrix proteins like collagen, fibronectin, proangiogenic factors. These are factors that will help recruit blood vessels to come in and start forming a capillary bed in this new wound tissue. And so they play a really good role for wound healing. And the problem is that there's a whole bunch of diseases where you start doing this wound healing process in a place where you really shouldn't. So, for instance, there's a disease called pulmonary fibrosis. And you can see this in coal miners, people who work in really dusty environments. There's just a little bit of irritation in the lungs. And if the lungs said, okay, a little bit of irritation, no big deal, everything would be fine. But the lungs overreact to this irritation. They think, oh, this is a wound. We're going to have to heal this wound. And they start bringing in all these wound healing cells to heal this perceived wound. Excuse me. And the lungs start filling up with scar tissue. So over here on the left, this is a section of a lung from a normal person. At least they were pretty normal until I guess they were probably like killed in a car accident or something. And this is the lung from a person with pulmonary fibrosis. And you can see all this scar tissue. And so over here on the left, all this white areas where the air can be moving in and out of the lungs. And here, there's no airspace over on the right. So the patient basically suffocates. And this wound healing process that shouldn't be occurring occurs all over the body to generate all kinds of fibrosing diseases. So keloids in the skin, fibrosis in the heart. This alone kills more people than cancer. Cirrhosis of the liver. That's why you guys shouldn't be drinking alcohol. You get fibrosis in the eye. You can get fibrosis in the kidney. Patients with diabetes, the kidneys will be damaged a little bit, but they're still working. And then they think, oh, this is a wound. And they start putting scar tissue into the kidney when they really shouldn't be. And that then causes the kidney to fail. So all of these ones with blue is where people have looked, and there's fibrocytes in there. All the ones in black, people haven't yet looked to see if there's fibrocytes, but there probably are. And collectively, these fibrosing diseases are associated with 30 to 45% of deaths in the US. And that compares to about 25% of deaths in the US coming from cancer. So fibrocytes play a good role for wound healing, but a bad role in these fibrosing diseases. But when we started working on this by accident, nothing was known about what regulates this differentiation. So if you take a healthy person and you scan all the way from the top of their head to the bottom of their feet, you probably won't see any fibrocytes in their body unless they've got a little cut that or a wound that they're healing. And you pretty much only see fibrocytes in a healing wound or in these fibrosing diseases. So now I'm going to move on to the next section and tell you how we found something that does regulate this differentiation. So we were doing a completely different experiment and working with human white blood cells. And when you work with human white blood cells, you normally grow them in a petri dish at 37 degrees in a broth that has sugars and amino acids. And you always throw in a little bit of blood serum, because blood serum is just full of all kinds of nutrients that the cells like. And you've probably seen in textbooks pictures of white blood cells. They're just little bitty round cells. For reasons that'll take hours to explain, we decided, oh, let's take some of these white blood cells and culture them in medium that doesn't have any serum. Turns out people never do this. And we took human white blood cells and put them into serum-free medium. And in about 3 days, we saw these huge long cells appearing in the culture. And turns out these are fibrocytes. If you take videos of these cells, you can see them trying to move and pull a wound closed, and they express markers that that you can see in fibrocytes in healing wounds and fibrotic lesions. But if you say, well, let's consider this to be the starting point, you get fibrocytes. And then when you add serum, you don't get fibrocytes, right? So this means that there's a factor in serum that inhibits the formation of these elongated cells that turn out to be fibrocytes. So we thought, well, if there's a factor in human blood serum that inhibits fibrocytes, maybe that could be useful as a therapeutic. So we got a bottle of human blood serum and started fractionating it. So probably in your undergraduate labs, you've done, say, some chromatography, or maybe by accident, you've done some chromatography. So if you take a piece of paper and you're writing on it with black ink and the paper gets wet, you'll see that as the water moves up, that you see the yellow and green and blue and different components of the ink will start separating. And if you haven't done it, just grab a piece of paper, make some ink marks. Black ink is good. Dip it into some water and you'll see the chromatography. So you can use chromatography to separate out all kinds of different things. And what we're looking at here is basically chromatography where you can separate out proteins. And what we've done is we've used different types of chromatography and different purification separation techniques to take human blood, fractionate it, and say which fraction contains the factor that inhibits fibrocyte differentiation. And we find that fraction, and turns out it'll contain like lots of protein. So we take that fraction and do further fractionation, and we finally got it to a point where, after fractionating with this technology, fractionating with that technology, we get a single protein that is the thing that's inhibiting fibrocyte differentiation. And we sent it off for analysis, and turns out to be a protein called serum amyloid P. It's made by the liver, and it's secreted into the blood. And if you look at people, the blood concentrations can vary quite a lot. And the concentrations are in the order of about 30 micrograms per mil, which is actually a fairly high protein concentration. And for the cognoscenti, this is not serum amyloid A, which does bad things, or amyloid precursor protein, which also does bad things, or amyloid beta, which also does bad things. Completely different. It just happens to have the word amyloid in its name. And people have made mice that where you've knocked out the gene that encodes serum amyloid P. And we're looking at the lungs of mice. Here are mice, normal wall-type mice. Here are mice that have been treated with a drug called bleomycin that induces fibrosis in the lungs. And you can see all this extra scar tissue material. This lung, the mouse is still alive, but it's going to be having a little bit of trouble breathing because there's so much scar tissue replacing the airways. If you do that same experiment with mice where you've knocked out the gene coding SAP, and you give them just a little bit of bleomycin, you see that there's a lot more scar tissue formation in the lungs. And so this indicates that a normal function of SAP is to limit fibrosis. If you look at patients with pulmonary fibrosis, you see that the average amount of SAP in the blood of pulmonary fibrosis patients is lower than controls. And if you parse out the data from these pulmonary fibrosis patients and you say, okay, let's plot the patient's SAP concentration, this particular patient's SAP concentration is against their lung air capacity, you see the patients with high levels of serum SAP, their lung air capacity is pretty high. If they were completely healthy, it would be 100%. And the patients with low levels of serum SAP, well, their lung air capacity is much lower than what it should be. And we see lower serum SAP levels in patients with kidney fibrosis, a weird skin fibrosis called scleroderma, and a bone marrow fibrosis called myelofibrosis. Okay, so now I'll tell you a little bit about how we can try and use SAP as a possible therapeutic. So if SAP is inhibiting the differentiation of monocytes to fibrocytes, one possibility is you can just prevent fibrosis by giving the patient SAP. So we used a mouse model. Oh wait, this is a rat model, excuse me, of pulmonary fibrosis. So you can take rats and look at their lungs. This is what a normal rat lung would look like, looking at sections. If you give a rat this drug bleomycin, it's a very standard model. You see that there's all this scar tissue formation, all this collagen in the lungs. And if you give them bleomycin and then treat with SAP, the lungs look basically normal. And if you don't give them bleomycin, you give them SAP, the lungs also look normal. And the cool thing is we can take rats, give them bleomycin, and then look at the blood oxygen levels with one of these, basically these pulse oximeters that in people you just put on a fingertip. And with bleomycin, the blood oxygen levels are abnormally low. But if you treat with bleomycin and say you wait 14 days to when there really is scar tissue forming, and then start treating with SAP, you get the blood oxygen levels come back. And we're looking at these lungs, the lungs of rats treated with bleomycin, and then we wait a while and then treat with SAP. The lungs look a lot better. So we started a company called it Promedier to develop SAP as a therapeutic. And Promedier went around to all these big cheese experts and said, hey, we'll give you some money and here's some SAP. Can you try this out in your model? And in all these models of fibrosing diseases in the eye, lung, kidneys, liver, heart, skin, GI system, SAP worked. So the company was able to raise a good bit of money, and something like $226 million. And that was able to fund clinical trials. And SAP worked in patients. So here we're looking at the decrease over 28 weeks of the lung air capacity. And in the controls, there's about a 5% decrease over 28 weeks. So pulmonary fibrosis is an unpleasantly progressive disease. And we can slow that progression quite a bit by treating patients with SAP, or it's also called pentraxin 2 or PRM151. This protein, unfortunately, has a whole bunch of different names. But the cool thing is the patients who were not so sick, so they hadn't started getting the current standard of care therapeutics, and so they're just starting to decline. When you treat those patients with pentraxin or SAP, you completely stop the decline in lung function. You just bang, they flatline. And the other cool thing is there's a very simple test to see how well the patients are doing in terms of their lung capacity. And you just say, how far can they walk in 6 minutes? And then you see over 28 weeks, how does that walk distance decline? So the walk distance in all the patients of the control group declined by about 33 meters over 28 weeks. In the patients that were being treated with SAP, or pentraxin 2, there was no decline. And the patients who weren't so sick and weren't getting a current standard of care, well, the placebo group, bang, you see a decline. But the patients treated with SAP, they actually improved. After 28 weeks, they could walk further. And this has never been seen with any therapeutic for pulmonary fibrosis. So this is really, really exciting. And this is why Roche bought the company and is currently enrolling patients in the phase 3 clinical trials. Okay, so now I'll finish up by telling you a little bit about the next generation therapeutics we're going after, involving identification of SAP receptor and SAP mimetics. So SAP has a good bit of similarity to a protein called C-reactive protein. And CRP is associated with heart disease and bad things. SAP is a good protein, CRP is a bad protein. And they have a lot of sequence similarity and a lot of structural similarity. So what we did was we mutated SAP amino acids that are different from CRP, that are on the surface of the protein, and said, will the SAP, that mutant SAP, will that inhibit fibrocyte differentiation? And none of these mutations did. And we were really baffled. And then we thought, what is it about SAP makes it different from CRP? And we thought and said, oh, wait a second. SAP has a post-translational glycosylation. So those of you who've had second year biology probably know that proteins, when they're being made in the endoplasmic reticulum for secretion, or they can, in some cases, get a chain of sugars put onto them. So SAP has this chain of sugars, CRP doesn't. And we can start cutting sugars off of SAP, and bang, when you cut sugars off of SAP, this data is really noisy, but basically you destroy the ability of the SAP to inhibit fibrocyte differentiation. So it looks like the sugar on the SAP is the really key thing that SAP is using to inhibit fibrocyte differentiation. So we said, well, what could be the receptor on monocytes? And so there's a protein called DC sign. It's a receptor. It looks for sugars, the kind of sugars that's on SAP, and it's involved in sort of calming down the immune system. So we did a bunch of work. I'm skipping over a lot of stuff to show that DC sign really is the receptor for SAP. But one cool thing from our studies is people had found small molecules that bind very strongly to the extracellular domain of DC sign. And we got 3 of them and asked, will these compounds inhibit fibrocyte differentiation? And all 3 of them do. There's this compound 1. Here's the structure. And it incredibly strongly and potently inhibits fibrocyte differentiation. So we tried this out. So we tried this out in a mouse model. And so what we've done here is we're looking at the lungs of mice. And here are control mice. Here are mice treated with bleomycin to induce pulmonary fibrosis. And then they were treated with DMSO, which is the vehicle that we dissolve the drug in. And they get pulmonary fibrosis. And then here are the lungs of mice that have been treated with bleomycin to induce pulmonary fibrosis. We wait 10 days and then start treating them with DMSO with a little bit of this compound 1. We sometimes call it 1866. And bang, the lungs look pretty much normal. And we can look at the collagen in these lungs, and with bleomycin, you get lots of collagen compared to control. And with different doses of this DC sign ligand, bang, you can shut down the fibrosis. You can strongly decrease the collagen in the lungs. So the next steps of the story are going to be presented to you by Tejas Kahardkar, who, as I mentioned, got his BSc in biotechnology from the amazing N.B. Mehta Science College. And so the conclusions of this part of the story is we noticed an odd behavior of cells led to a potential therapeutic. So for those of you who are just starting out your careers, you see something odd, follow it up. It changed our lives. And then we noticed this odd property of SAP. We were trying to mutate it, nothing would work. And that led to another set of potential therapeutics. The people who did this work are a guy named Daryl Pilling, who did that initial purification of SAP and the characterization. A whole large team of people did all those clinical trials. Two amazing graduate students, Nehemiah Cox, Jeff Crawford, did the work on those DC sign ligands. We used lots of human blood, and so I have to thank all the students who've donated blood for our work, and the phlebotomy staff who pull the blood out of the students, and of course the funding agencies, the Howard Hughes Medical Institute initially, and then the U.S. National Institutes of Health. So I'm going to stop there and say thank you very much.